Dear cryobiologists and students, I'm Yelda Özden Çiftçi from Gebze Technical University Department of Molecular Biology and Genetics from Turkey. I've been working with plant cryopreservation since 2005, and I would like to today share our results on determination of the potential of fructans in plant cryopreservation applications. Cryopreservation is a long-term storage of various plant materials in liquid nitrogen at very low temperatures where metabolic activity is triggered. And as metabolic activity is triggered, the maximum stability of phenotypic and genotypic trait is also ensured while plant genetic resources are preserved. For cryopreservation of plants, we can use different explant types like seeds, zygotic embryo, nodal buds, cell and callus cultures, somatic embryos, as well as seedlings, which we use in this study. There are different kinds of cryopreservation techniques, including dehydration, encapsulation dehydration, vitrification, encapsulation vitrification, and droplet vitrification and elsewhere. But the most used one is the vitrification, which contains as 5 basic steps, which can be summarized as cold acclimatization, pre-culturing, treatment with cryoprotective agents, storage in liquid nitrogen, thawing and post-thawing treatments. On this stage, cryoprotective agent exposure seems to be the most important one, and we use cryoprotective agents to eliminate deadly ice formation while cooling tissues and organs to cryogenic temperatures. In mammals, FPS and DMSO is used, but in plant cell we have several options, different plant vitrification solutions, among which PBS2 seems to be the most successful and the most applied one, which is developed by Sakai. It consists of glycerol, ethylene glycol, sucrose, and DMSO. While generally the other vitrification solutions is formulated just to decrease or eliminate DMSO in vitrification solution, as DMSO can be toxic to the plants. However, nowadays natural cryoprotectants seems to be also important for the cryopreservation studies. And one of these natural cryoprotectants are the fructans, which are known to protect plants against drought and cold stress in dry and harsh climatic conditions. And they are the most common non-structural carbohydrates after starch in nature. While starch biosynthesis comes to a halt at temperature below 10 degrees Celsius, the fact that the fructan biosynthesis is less affected by temperature provides a physiological advantage to the plant that can store fructan at low temperature. Therefore, the natural cryoprotectant fructan may have potential to be used in the cryopreservation studies. Thus, the aim of the study is to reveal out if utilization of different type of fructans in vitrification solution may help to have better survival in cryopreservation of Arabidopsis thaliana seedlings. So we use a optimized cryopreservation protocol for Arabidopsis, and we initially surface sterilize the seeds by using ethanol, sodium hypochlorite, and incubated in Mauritian sukuk medium without plant growth regulators. And the optimized protocol consists of usage of 48-hour in vitro germinated seedlings. So we also use this, and we osmoprotect by using glycerol and sucrose, and they hydrate with PBS2 at 0 degrees Celsius for 50 minutes, and then we rapidly cooled and rapid warmed, and we applied also 1.2 molar sucrose as a washing solution, and then recovery is carried out in MS medium, and we collect the data after 15 days of development, and we use 20 seedlings and repeated per each trial and repeated 3 times. And we used optimized protocol, but we modified with the usage of fructan in the PBS2 solution. So the control is just the same original PBS2, which contains 50% of DMSO. but the fructan group contains different types of fructans. In here we use microorganism derived or plant derived different kinds of fructans, and we incorporated 100 milligram per liter to 300 milligram per liter fructans to the individually to the PBS2 solution, and the DMSO concentration is reduced to 12.5 or 10% in the solution. When we come to the result, this is the first result that we obtain with the usage of different kinds of fructans, which we can see in the figure. And we applied 100 milligram per liter fructan for each, and we reduced the DMSO concentration to 12.5%. And in the control, in the non-cryopreserved ones, we can see that the graminan, HP inulin, and M611 resulted in the same the same survival with the original PBS2. But when we came to the cryopreserved ones, only graminan and HP inulin resulted in the statistically similar result with the original PBS2. With usage of other fructans resulted in lower survival in controls and also in cryopreserved ones. So this is the plants that we have. And so we concentrated in graminan and HP inulin, I mean two types of fructans, as they resulted best in the previous trials. And we reduced the DMSO concentration down to 10% now. And when we can see from the graphic that when the both tested fructans resulted in a decline in survival when we compare with the presence of 12.5% of DMSO. Also, we tried to improve our result by increasing the fructan concentration in the PBS2 solution. So DMSO concentration here is initially 12.5 and then to 10% individually, and you can see that even though we applied more fructan, the decline is still can be obtained in 12.5 and also 10% DMSO trials. So valid for the 200 milligram per liter graminan incorporation, and with the 300 milligram per liter fructan in comparison to the PBS2 solution for graminan and HP inulin. And this is in here we reduce the DMSO concentration to 12.5, and you can see that the result is lower than the 100 milligram per liter graminan or HP inulin incorporation to the PBS2 solution, and this result is lower when we decrease the DMSO concentration down to 10%. We also try to improve the vitrification solution by the inclusion of vitamin C as an antioxidant, because we know that the oxidative stress is very important stress that plant face during cryopreservation, and we used only one concentration according to the literature, and we can see also from the graph that the result is just the same to the control. In the control, the 50% DMSO is used, and but the morphologically the plant is better when the vitamin C is included to the PBS. So as a conclusion, according to the results, the highest recovery was observed with inclusion of 100 milligram per liter graminan and reduction of DMSO to 12.5% among the tested fructans. And the addition of vitamin C as an antioxidant to reduce oxidative stress during dehydration and freezing to this solution improved the morphological properties of the plant recovered after cryo. And the usage of fructans may have potential to be used in vitrification solution as a supplement in Arabidopsis thaliana. But of course, more trials should be done in order to valid this potential, such as we only use the fructans in the vitrification solution, but this can be also used in preculture medium or recovery medium. But it seems that there could be a potential of usage of natural cryoprotectants in the cryopreservation studies. Study is supported by Tribitak, and I would like to thank you for your attention.